Good morning, everyone. I hope you had a wonderful spring break, got a little rest, and I also hope that you are ready to come back for fourth quarter, jump in, get caught up, get some things done, and finish strong this school year. I'm excited for the things we're going to do. But let's go ahead and talk about this week. First, there's no school Monday, obviously, but for the rest of the week, let's look at what you're going to be doing. First, you're going to be looking at a Punnett Square Edpuzzle video on Google Classroom. Go through, work through that video. It shows you how to do a Punnett Square, which I'm also going to show you how to do in this video. So you're going to kind of see how to do it in two different places. The next assignment, there are some genetics questions. There's kind of some pictures that you're going to look at, and based on the pictures, you will answer the questions in the document. And one of the interesting things about this assignment is there's not necessarily a right or wrong answer. You're going to be making a lot of hypotheses. Like you're going to be taking some good guesses at what you think the answer should be. Because these questions are based more on, like I said, what you think it should be. Now, if you want to look some things up and help yourself answer the questions, that's fine. But the main point of this is to tell me what you think is going to happen. And that assignment is due at the end of the day, Thursday. And then finally, there's a Monohybrid Cross Punnett Square assignment. So here's where a couple things are going to differ a little bit. If you're in person, you will take the sheet and fill it out, work through it based on what you've learned in the Edpuzzle and my lesson here. If you are a remote virtual student, you're going to do the same exact lesson. The only difference is you're going to look at the document on the screen and then write it out on paper. And then you're going to take pictures of your paper and then submit it in Google Classroom to get the grade. That's the only difference. It's the same exact questions, the same exact Punnett squares. It's exactly the same. The only difference is how you'll turn it in to me. But yeah, let's go ahead and look at this week's lesson on monohybrid crosses and Mendelian genetics.
All right, we are going to talk a little bit about Gregor Mendel, who is considered the father of genetics. Let's begin. So the passing of traits to the next generation is called inheritance or heredity. And Gregor Mendel published his findings on the method of inheritance in garden pea plants. So he took these pea plants and started noticing that based on how they were kind of bred together, they looked different. And he started thinking, why do they look different? What's going on here? Well, cross-pollinated pea plants, which normally self-fertilize, he noticed that they rigorously followed various traits in the pea plants that he bred. So by studying this, he began the study of genetics, which again is the science of heredity. One trait that Mendel noticed was seed color. Some plants always produced green seeds, others always produced yellow seeds. And he crossbred the green and yellow seed plants, and then he called the green seed and yellow seed plants the parent generation or the pea generation. So the ones that you begin with are the parent generation or pea generation. As they were crossbred, they had their offspring, so to speak, and the offspring had offspring. These are the F1 and F2 generations. So the offspring of the pea cross-pollinated plants were the initial or first filial generation, or F1. The second filial generation, or F2, is the offspring of the F1. And in Mendel's peas, the green seed trait disappeared in the F1 generation, but reappeared in the F2 generation. So the F2 generation showed a ratio of 3-to-1 yellow to green seeds. So he noticed the first time the yellow and green bred together, only yellow came out of it. But then when he crossbred the yellow ones from that second generation, yellow and then some green appeared again. So he started to think, what happened? He only crossed yellow together, so why are there green? And that's what led to the next big question for him. So the F1 and F2 generations, during that, Mendel studied 7 different traits: seed or pea color, flower color, seed pod color, seed shape or texture, seed pod shape, stem length, flower position. And in all these cases, Mendel found that the F2 generations showed a 3-to-1 ratio of traits. No matter what, there was always a 3-to-1 ratio in that second offspring, which is the third generation or the F2 generation. So Mendel concluded that there must be two forms of the seed trait in the pea plant and that each was controlled by a factor. So the word allele, you're going to hear that a lot from now on. An allele is an alternative form of a single gene. So the gene for yellow seeds and the gene for green seeds are different alleles for the same gene. Now, alleles can either be what's called dominant or recessive. Dominant alleles control the traits that do appear in the F1 generation, that they're dominant, they're going to appear. Recessive alleles were masked in the F1 generation. And based on how the DNA came together is what determined whether they were going to express the dominant or recessive traits. And we'll see a little bit about that in a second. So dominance, when modeling inheritance, the dominant allele is represented by a capital letter. We're going to use Y here. And a recessive allele is represented with a lowercase letter, and in this case, a lowercase y. An organism with two of the same alleles for a particular trait is called homozygous for that trait. So either two big Ys or two little y's. If the traits are the same allele, it's homozygous. An organism with two different alleles for that trait is heterozygous. So big Y, little y, they are different alleles for the same trait. In heterozygous individuals, the dominant trait is going to be observed. So obviously in homozygous, if you have two capitals for homozygous, it's going to be dominant. If you have two lower cases, it's going to be recessive. And if you have a capital and a lowercase, the dominant takes over and will be observed. So heterozygous is always showing the dominant trait. And then we have the words genotype and phenotype. So the appearance of an organism does not always indicate which pair of alleles it possesses. And an organism's allele pairs are called the genotype. So genotype are the genes, like what pair of genes do they have? Now the observable characteristics or the outward expression of the allele pair is the phenotype. So when we go back to those seed pea plants, they used Y for yellow. So big Y would be the genotype, and yellow would be the phenotype. The observable trait is the phenotype. So the genotype is the genes. So big Y. Phenotype is what you see. So yellow. Now Mendel's law of segregation states that the two alleles for each trait separate during meiosis. So if you remember during meiosis, we saw those chromosomes pull apart. The two alleles are separated. During fertilization, two alleles for that trait unite. Heterozygous organisms are called hybrids. So when those things come together, we call that a hybrid. Now, when we start to cross the genetics in this, and we're looking at it in terms of these pea plants here, a monohybrid cross is a cross that involves hybrids for a single trait. And a dihybrid cross is the simultaneous inheritance of more than one trait in the same plant. So monohybrid crosses, we're just talking about one specific trait. Dihybrid crosses, we're talking about multiple traits at the same time. Now, we also have what's called the law of independent assortment. This law states that there's a random distribution of alleles occurring during gamete formation. So when your gametes are being formed, there's kind of no rhyme or reason as to why the genes are distributed the way they are. There's so many possible combinations that it's completely random and you can't really pinpoint what's going to happen, which ones are going to be given. Genes on separate chromosomes sort independently during meiosis, and each allele combination is equally likely to occur. Like, it's just as probable to get any of these as it is the other. Now, Punnett squares are what we can use to predict possible offsprings of a cross between two genotypes. So if we have like the ability to roll your tongue, some people can do that. And when I say roll your tongue, I mean do this. Some people can do that. Some people cannot do that. That's actually a genetic trait. So we're going to do capital T for the ability to be able to do that, and lowercase t for the inability to do that. And we're going to cross two heterozygous individuals. on the top of the box and on the left side of the box. And basically you cross multiply and put the results inside the box. So big T times big T, you get two big T's. Big T times little t, you get big T little t. Big T times little t, you get big T little t. And little t, you get little t little t. So you can see here, homozygous dominant is always dominant. Heterozygous is always dominant. And then homozygous recessive is the only recessive. So that's why it's 3-to-1 no matter what, which we said earlier. So the number of squares is determined by the number of different types of alleles produced by each parent. Typically, we're just going to have 4 squares here. Now, in a dihybrid cross, things get a little more complicated. You don't have to worry too much about this. I'm not going to make you do any of these. Next week, you probably will do a regular 4-squared Punnett square. But for now, this dihybrid cross, I'm just going to explain what it is. So we have 4 types of alleles from the male gametes and 4 types of alleles from the female gametes that are produced. And if you look through it, it produces a ratio of 9-3 to 3-to-1. So 9 of one kind, 3 of one, 3 of one, and 1. Now, probability, when we're talking about probability, the inheritance of genes can be compared to the probability of flipping a coin. Actual data might not perfectly match predicted ratios. If you flipped a coin 50 times, you might get heads every time.
The chili cook-off will begin in 8 minutes, and we ask that you please send your email. Thank you.
If you flip a coin 50 times, you could get heads 50 times, or you can get tails 50 times, or you could get heads 10 times and tails 40 times. Like, that's actually how it works with genetics. It's random. But we are able to take mathematical ratios to figure out the probability, like what might be extremely possible to happen. And Mendel's results were not exactly a 9-3 to 3-to-1 ratio, but the larger the number of the offspring involved, the more likely it will match the results predicted by a Punnett square. That's why using something like pea plants is good, because you get a whole bunch of them. With humans, it's not exactly the same, or animals, because typically you get 1 offspring. With animals, you might get more than 1, but it would take a lot longer and a lot more time and effort to study that in a human. That's why something like this is wonderful to study with something like a pea plant. Now, genetic recombination, the new combination of genes produced by crossing over and independent assortment is called genetic recombination. Combination of genes due to independent assortment can be calculated using the formula 2 to the nth power, where n is the number of chromosome pairs. So any possible of male gamete can fertilize any possible of female gametes. So the possible combinations after fertilization are 2 to the nth power times 2 to the nth power. So humans have 23 pairs of chromosomes, so 46 chromosomes. A male gamete gives 23 and a female gamete gives 23. So in humans, that would be 2 times the 23rd power, or excuse me, 2 to the 23rd power times 2 to the 23rd power to get you the all possible combinations after fertilization in a human. It's a very large number. Now, gene linkage, genes located close to each other on the same chromosome are said to be linked. They usually travel together during gamete formation, and gene linkage results in an exception to Mendel's law of independent assortment. If you look through these pictures here, they've kind of used color to indicate where similar genes are. And you can see that the colors are lined up in each of these. So as they travel, those similarly linked genes are going to travel the same way. When centromeres separate and gametes form, the same type of genes are going to go together so that later they can be lined up again similarly. We also have what's called chromosome maps. Crossing over occurs more frequently between genes that are farther apart. Crossover data can be used to create chromosome maps, which are depictions of how genes are arranged on a chromosome. Right here we have different body and eye colors amongst flies, and they've labeled them with colors to kind of show some chromosome maps here, which are the directions of how those genes might be arranged. And now we have what's called polyploidy. So this is the occurrence of one or more extra sets of all chromosomes in an organism. A triploid organism is designated 3N, which means that it has 3 complete sets of chromosomes. So you would take 3 times however many sets they have. Many agricultural crops are polyploid. Wheat is 6N, oats is 6N, sugarcane is 8N. Polyploid plants often have increased vigor and size. So in plants, this is a great thing. If you are farming and you want to yield more, bigger, better crops, this is actually great. It's not so great in humans or animals, though.
Okay, now that you have taken a look at that information, let's look at how to work through Punnett squares, some monohybrid Punnett squares. I'll do a couple examples for you. And yes, these are the exact examples from your assignment. So the first couple, you're going to see how to do them. Now let's read through these and look at some of the important words here, and we'll walk through what you should do. Okay, the first scenario says, in humans, brown eyes are dominant over blue eyes. What type of offspring would you expect if you crossed a heterozygous brown-eyed person to a heterozygous brown-eyed person? Well, first of all, we can see both people that are crossing are heterozygous brown-eyed people. So we're going to have the same thing on the top of the square as we do on the left side of the square. Now, heterozygous is the important term here. Hetero means different. So that means our alleles are going to be different. And if you remember from the lesson, if it is a dominant allele, it gets a capital letter. If it is a recessive allele, it gets a lowercase letter. Dominant can be either 2 ways. It can be 2 capital letters or a capital letter and a lowercase letter, because even when it's paired with a recessive, the dominant still takes over, so it still counts as being dominant. So if it is dominant, it's either capital, capital, or capital lowercase. If it is recessive, it is lowercase, lowercase. That's the only way it can be recessive. Another thing to note, the letters aren't given to you in these questions, so you get to make the letters up. And honestly, it does not matter what letters you pick, as long as you make the right combination of capital letters and lowercase letters. So in this case, we're talking about brown eyes. So I'm going to use the letter B. Now, heterozygous means it's different. So capital, lowercase. And we cross a heterozygous brown-eyed person with another heterozygous brown-eyed person. And heterozygous means different. So it is going to be big B, little b, crossed with big B, little b, because we have 2 of the exact same, heterozygous brown. And what you do, it's kind of like cross multiplying with the letters. Big B goes here. Big B, little b goes here when we cross those. Big B, little b again. And little b again. So this is what you could expect. And over here, we're going to write down the different types of genotypes. We have big B. We have big B, little b. We have another big B, little b. And we have a little b. And we can see that this one is homozygous dominant. These 2 are heterozygous dominant. And this one is homozygous recessive. Now we have 1, 2, 3 chances to be dominant and 1 chance to be recessive. But these are the genotypes. The genotypes are the letters, basically. What are the genes? These are the genotypes. Over here, for phenotypes, this is what are you going to see? You are going to either see the dominant or the recessive. Okay, for phenotype, let's look at our different probabilities here. We know that if we have 1, 2, 3 possible dominant chances of having heterozygous or homozygous brown eyes, That's a 75% chance that we have brown eyes. That leaves us with a 25% chance of having what the question said was the recessive one, and that's blue eyes. So if you look here, the genotypes are just these written out letters. Phenotypes are what you're actually going to physically see and what are the probabilities of seeing that. Let's do another one here. Number 2 says a widow's peak hairline is dominant to a straight hairline. A widow's peak's like when you have that hair that comes down in the front, and that's going to be dominant to a straight across hairline. I don't even have a hairline, so what am I talking about? But anyway, if we look here, let's pick maybe the letter H because we're talking about different hairlines. And here's what we're going to cross. Heterozygous widow's peak to a straight hairline. Now, heterozygous means it's going to be dominant but different. So that would be big H, little h. So big H, little h. Now, the straight hairline is recessive, and the only way to have recessive is 2 lowercase. So little h, little h. And once we have that, we can start to fill it in. We're going to have big H, little h here. We're going to have little h here. We're going to have big H, little h here. And we're going to have little h here. So when we go to write our genotypes, we're going to have big H, big H, little h, little h, little h, little h, little h. So we can see now it's 50-50. So with our phenotypes here, it's a 50% chance of having widow's peak. And it's also a 50% chance of having straight hair. Okay. So again, just note, genotypes are the letters, the alleles, and phenotypes are what you physically see and how probable it is to see that particular trait. Okay, so what I want you guys to do is go through the rest of this. Again, if you're in person, just fill it out on the sheet. If you're remote, draw out your boxes and genotypes and phenotypes on a piece of paper, take a picture of that paper, and then submit it on Classroom. And now I want you guys to go ahead and work on the assignments for the week. As usual, if you have any questions, please send me an email and I will answer your questions. If you're in person, raise your hand and ask me. Either way, please reach out if you need some help. Otherwise, good luck on these assignments. Good luck on catching up on some things you might need to catch up on. And let me know if you need anything.